Section 50 of Gray's Anatomy, Part 4. This is a LibriVox recording. All LibriVox recordings are in the public domain. For more information or to volunteer, please visit LibriVox.org. Recording by Morgan Scorpion. Anatomy of the Human Body, Part 4, by Henry Gray. Tunics of the Eye, Part 1. From without inward, the three tunics are: 1. A fibrous tunic, consisting of the sclera behind and the cornea in front. 2. A vascular pigmented tunic, comprising, from behind forward, the choroid, ciliary body, and iris. And 3. A nervous tunic, the retina. The fibrous tunic, tunica fibrosa oculi. The sclera and cornea form the fibrous tunic of the bulb of the eye. The sclera is opaque, and constitutes the posterior 5/6 of the tunic. The cornea is transparent, and forms the anterior 1/6. The sclera. The sclera has received its name from its extreme density and hardness. It is a firm, unyielding membrane, serving to maintain the form of the bulb. It is much thicker behind than in front. The thickness of its posterior part is 1 mm. Its external surface is of white color, and is in contact with the inner surface of the fascia of the bulb. It is quite smooth, except at the points where the recti and obliqui are inserted into it. Its anterior part is covered by the conjunctival membrane. Its inner surface is brown in color and marked by grooves, in which the ciliary nerves and vessels are lodged. It is separated from the outer surface of the choroid by an extensive lymph space, spatium perichoroideal, which is traversed by an exceedingly fine cellular tissue, the lamina suprachoroidea. Behind it is pierced by the optic nerve, and is continuous through the fibrous sheath of this nerve with the dura mater. Where the optic nerve passes through the sclera, the latter forms a thin cribriform lamina, the lamina cribrosa sclerae. The minute orifices in this lamina serve for the transmission of the nervous filaments, and the fibrous septa dividing them from one another are continuous with the membranous processes which separate the bundles of nerve fibres. One of these openings, larger than the rest, occupies the centre of the lamina. It transmits the central artery and the vein of the retina. Around the entrance of the optic nerve are numerous small apertures for the transmission of the ciliary vessels and nerves, and about midway between this entrance and the sclerocorneal junction are 4 or 5 large apertures for the transmission of veins. Venae vorticosae. In front, the sclera is directly continuous with the cornea, the line of union being termed the sclerocorneal junction. In the inner part of the sclera, close to this junction, is a circular canal, the sinus venosus sclerae, canal of Schlemm. In a meridional section of this region, this sinus presents the appearance of a cleft, the outer wall of which consists of the firm tissue of the sclera, while its inner wall is formed by a triangular mass of trabecular tissue. The apex of the mass is directed forward, and is continuous with the posterior elastic lamina of the cornea. The sinus is lined by endothelium and communicates externally with the anterior ciliary veins. The aqueous humour drains into the scleral sinuses by passage through the pectinate villi, which are analogous in structure and function to the arachnoid villi of the cerebral meninges. Structure. The sclera is formed of white fibrous tissue intermixed with fine elastic fibres. Flattened connective tissue corpuscles, some of which are pigmented, are contained in cell spaces between the fibres. The fibres are aggregated into bundles, which are arranged chiefly in a longitudinal direction. Its vessels are not numerous, the capillaries being of small size, uniting at long and wide intervals. Its nerves are derived from the ciliary nerves, but their exact mode of ending is not known. The cornea. The cornea is the projecting, transparent part of the external tunic, and forms the anterior 1/6 of the surface of the bulb. It is almost circular in outline, occasionally a little broader in the transverse than in the vertical direction. It is convex anteriorly and projects like a dome in front of the sclera. Its degree of curvature varies in different individuals, and in the same individual at different periods of life, being more pronounced in youth than in advanced life. The cornea is dense and of uniform thickness throughout. Its posterior surface is perfectly circular in outline, and exceeds the anterior surface slightly in diameter. Immediately in front of the sclerocorneal junction the cornea bulges inward as a thickened rim, and behind this there is a distinct furrow between the attachment of the iris and the sclerocorneal junction. This furrow has been named by Arthur Thomson the sulcus circularis corneae. It is bounded externally by the trabecular tissue already described as forming the inner wall of the sinus venosus sclerae. Between this tissue and the anterior surface of the attached margin of the iris is an angular recess, named the iridial angle or filtration angle of the eye. Immediately outside the filtration angle is a projecting rim of scleral tissue which appears in a meridional section as a small triangular area, termed the scleral spur. Its base is continuous with the inner surface of the sclera immediately to the outer side of the filtration angle, and its apex is directed forward and inward. To the anterior sloping margin of this spur are attached the bundles of trabecular tissue just referred to. From its posterior margin the meridional fibres of the ciliaris muscle arise. Structure. The cornea consists from before backward of 4 layers, viz. 1. The corneal epithelium, continuous with that of the conjunctiva. 2. The substantia propria. 3. The posterior elastic lamina, and 4. The endothelium of the anterior chamber. The corneal epithelium, epithelium corneae anterior layer, covers the front of the cornea and consists of several layers of cells. The cells of the deepest layer are columnar, then follow 2 or 3 layers of polyhedral cells, the majority of which are prickle cells similar to those found in the stratum mucosum of the cuticle. Lastly, there are 3 or 4 layers of squamous cells with flattened nuclei. The substantia propria is fibrous, tough, unyielding, and perfectly transparent. It is composed of about 60 flattened lamellae superimposed one on another. These lamellae are made up of bundles of modified connective tissue, the fibres of which are directly continuous with those of the sclera. The fibres of each lamella are for the most part parallel with one another, but at right angles to those of adjacent lamellae. Fibres, however, frequently pass from one lamella to the next. The lamellae are connected with each other by an interstitial cement substance, in which are spaces, the corneal spaces. These are stellate in shape, and communicate with one another by numerous offsets. Each contains a cell, the corneal corpuscle, resembling in form the space in which it is lodged, but not entirely filling it. The layer immediately beneath the corneal epithelium presents certain characteristics which have led some anatomists to regard it as a distinct membrane, and it has been named the anterior elastic lamina. Lamina elastica anterior limiting layer, Bowman's membrane. It differs, however, from the posterior elastic lamina in presenting evidence of fibrillar structure, and in not having the same tendency to curl inward or to undergo fracture, when detached from the other layers of the cornea. It consists of extremely closely interwoven fibrils, similar to those found in the substantia propria, but contains no corneal corpuscles. It may be regarded as a condensed part of the substantia propria. The posterior elastic lamina, lamina elastica posterior, membrane of Descemet, membrane of Demors, covers the posterior surface of the substantia propria, and is an elastic, transparent, homogeneous membrane of extreme thinness, which is not rendered opaque by either water, alcohol, or acids. When stripped from the substantia propria it curls up, or rolls upon itself with the attached surface innermost. At the margin of the cornea the posterior elastic lamina breaks up into fibres which form the trabecular tissue already described. The spaces between the trabeculae are termed the spaces of the angles of the iris, spaces of Fontana. They communicate with the sinus venosus sclerae and with the anterior chamber at the filtration angle. Some of the fibres of this trabecular tissue are continued into the substance of the iris, forming the pectinate ligament of the iris, while others are connected with the forepart of the sclera and choroid. The endothelium of the anterior chamber, endothelium camerae anterioris, posterior layer, corneal endothelium, covers the posterior surface of the elastic lamina, is reflected onto the front of the iris, and also lines the spaces of the angle of the iris. It consists of a single stratum of polygonal flattened nucleated cells. Vessels and nerves. The cornea is a non-vascular structure. The capillary vessels ending in loops at its circumference are derived from the anterior ciliary arteries. Lymphatic vessels have not yet been demonstrated in it, but are represented by the channels in which the bundles of nerves run. These channels are lined by an endothelium. The nerves are numerous and are derived from the ciliary nerves. Around the periphery of the cornea they form an annular plexus, from which fibres enter the substantia propria. They lose their medullary sheaths and ramify throughout its substance in a delicate network, and their terminal filaments form a firm and closer plexus on the surface of the cornea proper, beneath the epithelium. This is termed the subepithelial plexus, and from it fibrils are given off which ramify between the epithelial cells, forming an intraepithelial plexus. The vascular tunic, tunica vasculosa oculi. The vascular tunic of the eye is formed from behind forward by the choroid, the ciliary body, and the iris. The choroid invests the posterior 5/6 of the bulb, and extends as far forward as the ora serrata of the retina. The ciliary body connects the choroid to the circumference of the iris. The iris is a circular diaphragm behind the cornea, and presents near its centre a rounded aperture, the pupil. The choroid, choroidea. The choroid is a thin, highly vascular membrane of a dark brown or chocolate colour, investing the posterior 5/6 of the globe. It is pierced behind by the optic nerve, and in this situation is firmly adherent to the sclera. It is thicker behind than in front. Its outer surface is loosely connected by the lamina suprachoroidea with the sclera. Its inner surface is attached to the pigmented layer of the retina. Structure. The choroid consists mainly of a dense capillary plexus, and of small arteries and veins carrying blood to and returning it from this plexus. On its external surface is a thin membrane, the lamina suprachoroidea, composed of delicate non-vascular lamellae, each lamella consisting of a network of fine elastic fibres along which are branched pigment cells. The spaces between the lamellae are lined by endothelium, and open freely into the perichoroidal lymph space, which in its turn communicates with the periscleral space by the perforations in the sclera through which the vessels and nerves are transmitted. Internal to this lamina is the choroid proper, consisting of two layers, an outer composed of small arteries and veins with pigment cells interspersed between them, and an inner consisting of a capillary plexus. The outer layer, lamina vasculosa, consists in part of the larger branches of the short ciliary arteries which run forward between the veins, before they bend inward to end in the capillaries, but is formed principally of veins, named, from their arrangement, the venae vorticosae. They converge to 4 or 5 equidistant trunks, which pierce the sclera about midway between the sclerocorneal junction and the entrance of the optic nerve. Interspersed between the vessels are dark, star-shaped pigment cells, the processes of which, communicating with those of neighbouring cells, form a delicate network or stroma, which toward the inner surface of the choroid loses its pigmentary character. The inner layer, lamina choriocapillaris, consists of an exceedingly fine capillary plexus, formed by the short ciliary vessels. The network is closer and finer in the posterior than in the anterior part of the choroid. About 1.25 cm behind the cornea its meshes become larger, and are continuous with those of the ciliary processes. These two laminae are connected by a stratum intermedium consisting of fine elastic fibres. On the inner surface of the lamina choriocapillaris is a very thin, structureless, or faintly fibrous membrane, called the lamina basalis. It is closely connected with the stroma of the choroid, and separates it from the pigmentary layer of the retina. One of the functions of the choroid is to provide nutrition for the retina, and to convey vessels and nerves to the ciliary body and iris. Tapetum. This name is applied to the outer and posterior part of the choroid, which in many animals presents an iridescent appearance. The ciliary body, corpus ciliari. The ciliary body comprises the orbiculus ciliaris, the ciliary processes, and the ciliaris muscle. The orbiculus ciliaris is a zone of about 4 mm in width, directly continuous with the anterior part of the choroid. It presents numerous ridges arranged in a radial manner. The ciliary processes, processus ciliaris, are formed by the inward folding of the various layers of the choroid, i.e. the choroid proper and the lamina basalis, and are received between corresponding foldings of the suspensory ligament of the lens. They are arranged in a circle and form a sort of frill behind the iris, around the margin of the lens. They vary from 60 to 80 in number, lie side by side, and may be divided into large and small. The former are about 2.5 mm in length, and the latter, consisting of about one third of the entire number, are situated in spaces between them, but without regular arrangement. They are attached by their periphery to 3 or 4 of the ridges of the orbiculus ciliaris, and are continuous with the layers of the choroid. Their opposite extremities are free and rounded, and are directed toward the posterior chamber of the eyeball and circumference of the lens. In front they are continuous with the periphery of the iris. Their posterior surfaces are connected with the suspensory ligament of the lens. Structure. The ciliary processes are similar in structure to the choroid, but the vessels are larger and have chiefly a longitudinal direction. Their posterior surfaces are covered by a bilaminar layer of black pigment cells, which is continued forward from the retina, and is named the pars ciliaris retinae. In the stroma of the ciliary processes there are also stellate pigment cells, but these are not so numerous as in the choroid itself. According to Henderson, the aqueous humour is a secretion formed by the active intervention of the epithelial cells lining the apices of the ciliary processes. End of section 50.